Hi, I'm Sara, and I have to say, first of all, that I am so honored to be here speaking to all of you. You all make such a tremendous impact on breast cancer and breast cancer advocacy, and I really never imagined that I would get to speak to all of you. So it's really just an amazing, amazing day. But I am very involved in cancer research, and I like to think about it, really, as a puzzle in a darkened room. Only a few areas are lit, and we can only understand pieces of what seems at times to be an infinitely complicated, interwoven mystery. Science has always fascinated me. I spent years in the science section at my local public library, and I decided I wanted to be a geneticist in 5th grade when I first learned about mutation. But my enthusiasm for experimentation only grew as I learned about everything from rocks and minerals to Newtonian laws and gravitational motion. Above all, I love science's limitless potential because there are thousands of different questions that can be asked, and there is an ever-expanding set of experiments that can be used to find the answers. When I was 9, I lost my close cousin Gail to metastasis. And when I was 9, I didn't know what metastasis meant, but all I felt was the impact cancer had already on my family. And I began learning about cancer research, real research, in 7th grade in time for a science project. But it took months of highlighting dictionaries, Google searches, and the very helpful source of Wikipedia to understand the unbelievable work that researchers are doing, in many cases across the globe, on breast cancer metastasis. And as soon as I started to even comprehend really what they were on the verge of discovering, or in some cases had even discovered, I dreamt of working with them in their laboratory and learning the techniques and tools that you need for research, and really the techniques and tools you need to make an impact alongside these amazing scientists in the laboratory. So the minute I turned 16 and I was legally allowed to work in a cancer research laboratory in New York City, I started emailing people. And when I was 16, I didn't even know what the word no meant, so the many, many rejections never really bothered me. I emailed over 150 scientists, and 2 of them said maybe. And eventually I ended up with my mentor, the amazing, beyond talented mentor, Dr. Vivek Mittal at Weill Cornell Medical College. And after that, things really started falling into place. I was lucky to be trained in the basics of in vitro cell culture by a fantastic postdoctoral research fellow, and I had a fearless mentor who was willing to entrust the most complicated procedures to an enthusiastic yet completely untrained teenager. The lab started for me as a summer internship, but as I fell in love with research and I became so dedicated to my project, I kept going during the year and throughout the subsequent summers, and it really became a permanent part of my life. I developed a routine during the school year: 90 minutes of cell culture in the morning, 4 to 5 hours of experimentation after school. And slowly, as I compiled more and more data from many different experiments, I started seeing a story come into play. My research focuses on epigenetic modifications in breast cancer metastasis. Epigenetics is a relatively new field in cancer research that's focused on the mutations to genetic material that are not incorporated into the DNA sequence. These modifications can turn off necessary tumor suppressor genes and thus permit the onset of cancer and cancer progression. Cancer can originate from different mutations that happen in cells' genetic material, and there are several well-known mutations in breast cancer. So many major therapies today target these specific mutations to halt the growth of the primary tumor. But it's not just about the primary tumor, because it's only when breast cancer metastasizes that it becomes a devastating and incurable disease. Breast cancer is one of the most highly publicized cancers, and a stunning amount of money, billions of dollars, is funneled into research every single year. So little is shared about metastasis. And when women progress to late-stage breast cancer, it seemed to me that they practically fell off the ever-present pink ribbon. Only in recent years have researchers begun to delve into the intricate interactions and pathways that lead to metastasis. Some of the pathways that are studied are actually epigenetic switches that have been correlated with poor prognosis and worse disease-free survival. My research focuses on one such switch. Normally, critical genes can prevent the onset of cancer. In breast cancer metastasis, a specific group of proteins can turn off the genes that normally prevent cancer growth and progression. I used a new chemical to block the activity of these cancer-inducing proteins called the polycomb group proteins, and I found that blocking this activity seemed to suppress the aggressive characteristics of breast cancer metastasis. But for me, the most exciting part of my research has been my work on cancer stem cells. The latest research on tumor biology is changing. The classically held original theory on tumor evolution begins with mutations occurring in a small number of cells that then leads to a domino effect of uncontrolled growth and proliferation. Some researchers believe that only a small group of tumor cells had the tumorigenic ability to seed and initiate tumors, but other scientists believe that all tumor cells had the same tumorigenic ability. And while novel work on the field has recently focused on a specific subset of tumor cells called cancer stem cells, basically tumor cells that have these added stem-like mutant properties, like the ability to self-renew indefinitely and practically become immortal. This is the group of cells that may be responsible for cancer initiation, tumor progression, metastasis, and even recurrence and chemoresistance. Data has been published on both sides of the cancer stem cell debate, and it is a theory that's constantly evolving and growing. But data does exist, and it has strong clinical relevance. One research group found that after chemotherapy or radiation, the cancer stem cell population in patients actually increased. But to date, there is not a single therapy that's used clinically to suppress cancer stem cells. I found that the blockade of the protein pathway I studied seems to block the breast cancer stem cell population. I'm in the middle of a paper submission process, but we're hoping that this research could directly contribute to a clinical trial that would help patients with late-stage metastatic disease. Every piece of data seems to be fitting into a puzzle where a small subset of tumor cells is responsible for tumor initiation and upkeep. These cells are resistant to chemotherapy and are resistant to other common early-stage treatments like radiation. Every year, hundreds of compounds enter clinical trials, but the primary endpoint in these trials is often progression-free survival, which is usually based on tumor measurements. But by confining outcomes to these narrow definitions, the trials could be missing therapies that wouldn't significantly impact the primary tumor size, but could treat or prevent metastasis by targeting the cancer stem cell population. Actually, I think the best possible therapy for patients would be a combination between therapies targeting bulk tumor cells, like normal standard chemotherapies used today, and specific therapies aimed at targeting the cancer stem cell population and eliminating that population. Because not only would that combination therapy treat the patient's current stage of cancer, but it would also eliminate the threat for resistance, further spread, or recurrence. My research has been an incredible journey with thousands of hours in the lab, 100 experiments that worked, and hundreds more that dramatically failed. And also a lot of new techniques both to learn, to create, and to modify. But I really have to thank the scientists who taught me the basics of science, but even more importantly, taught me the persistence, patience, and open-mindedness that's critical for science research, and especially research with direct clinical connections. This year, I had the amazing opportunity to participate in a national high school competition called the Intel Science Talent Search. And I remember a moment when one of my friends from Intel said, "It doesn't matter where you came from or who you are, it just matters what you know and what you want to discover." I'm 18, and when I began this journey, I never imagined it would bring me to this point, sharing the stage and attending such a fantastic leadership summit. I think science isn't about the unsocial geek wearing the lab coat with a bubbling test tube in the closet laboratory. I don't think it's a divide between scientists and the public, the clinic, or the laboratory. It's not even about the number of papers you publish or the patents you have. It's really about the lives that you save, the side effects you can prevent, and the mysteries of cancer and mutation that you can unravel. I think we can only get to this point. and achieve the amazing goal to know how to end breast cancer by 2020, if scientists, clinicians, citizens, congressmen, advocates, patients, and survivors all band together, alone, we would seem isolated in the clinic, the office, the laboratory. But together, we can fight back, stop cancer, and find a cure. I'm so honored to be at the 2014 National Breast Cancer Coalition Leadership Summit with all of you, my breast cancer heroes, and I'm so excited for the next 3 days. Thank you so much.